All right. So is everybody that we're expecting here?
Yeah.
Awesome. Cool. Well, it's so nice to see you all. My name is Lizzie. I may have met a couple of you who were possibly at our lab experience program last year. Really excited to have you all here with us again for Lab Backstage. Lab Backstage program was kind of born out of our lab experience program in the hopes of connecting scientists to students in middle and high school to about the work that they do and also to talk about sort of who they are and how they got into science and conveying the human perspective of all of this. And so it's kind of informal in that way. We invite you all to ask questions either in the chat or through raising your hand and speaking. And yeah, really excited to have you all here to learn with us. So our speaker for this lab backstage is Iram, and she will be telling you guys all about the awesome research that she does. So without anything further, I'll pass it along to Iram.
Hello, thank you, Lizzie, for the introduction. And my name is Iram. I am hoping today to show you our laboratory and a little bit talk about the research that we are doing in our lab. But before kind of going to the science part, I kind of first want to introduce myself. And as I said, my name is Iram, and I am from Turkey. I grew up in Istanbul. Um, Sorry, my phone is making an echo. I'm just gonna leave the meeting. Okay, it is distracting me otherwise. Okay, so yeah, I'm from Turkey. I grew up in Istanbul, and I did all my undergrad studies in Istanbul in Turkey. And I become interested in science, I guess, in middle school, but mostly in high school. I always enjoy my biology classes, and in middle school, like my science teacher, he was great, and he actually like we did like a small project in lab together. Actually, the only thing I did was kind of holding a beaker. I really did nothing, but it was still so much fun. And in high school, but I mean, like that, I guess in middle school with that experience, kind of already knew that I like biology, I like learning about the environment, but I kind of didn't know how to become a scientist because there is no scientist in my family. My parents, they didn't go to like grad school. So I didn't have an idea that like, oh, when I grow up, I want to be scientist. Like I wasn't thinking about that. But in high school, at the end of high school in Turkey, we take this exam. And based on your exam score, you choose like a department in university and also the which university that you want to go. And when I was like in high school, I was searching for different majors. And then one thing I see was like molecular biology. Again, I had no idea what molecular biology is. And then I just like Google it and like read blogs of like students studying molecular biology and try to learn a little bit about it. And then I visited the universities and asked like the students in person and also the teachers in the universities in person. That's basically how I kind of decided that, okay, that's really what I want to do. Even then, like I actually really didn't know that how to become like a scientist. As I said, because I really didn't have an example. But then during my college, I actually had the chance to do internship. I start first like working in laboratories in my own university, but also in summer then I had the chance to go United Kingdom and I had a chance to do summer research at University of Cambridge. And there are actually very cool like summer programs and scholarship opportunities for college students. That's how I visited England. And during my summer research internship, I actually decided, okay, I want to do like my PhD and I want to become like scientist and I want to do research for my, uh, I want to do research basically. And yeah, and now I'm here at Rockefeller University. I am a third year grad student. And this is actually, as I said, I grew up in Istanbul. So this is I came United States for grad school. I haven't been here before. And I still feel like I'm a tourist. I really love New York City. I love walking around the city, like discovering the neighborhoods. I am a food person. I enjoy trying like different foods. And I kind of have this fun poll question that I want to ask you so that we kind of get know each other better. And I want to ask you, what is your favorite New York stew foods? And you guys can like choose the options.
So for those of you who are in a classroom, you can put your answers in the chat if one of you wants to just like add them and we can read them out too.
Okay, so tell me one thing and I'll put it in. Pizza? Yeah.
Also, feel free to like, if you have any recommendation of any place, feel free to write the chat. As I said, I still feel like I'm a tourist in New York, so I would love to go there and try.
Soup? Seriously, I'm all food. Yeah, soup.
Soup.
Okay, Raquel, what do you like?
I forgot that soup. Sorry.
Sarah, what do you like? Sarah, what do you like? Pizza? Who's next? Rebecca, what do you like? What? Sushi? Who says home cooked? Abby, what do you like? What do you like, Alana? What do you like? What? What's your favorite food? You like everything? Okay.
Alana likes acai bowls.
What?
She likes acai
Oh,
bowls.
okay. I know, but I don't, if I backspace it, closes for him.
Yeah, it is difficult to choose. I like every food too.
What do you like? Pasta? Copycat.
So while everybody is
Wow.
adding in their answers, I shared
Yeah.
the poll responses.
Yeah, there are many pizza people. I love pizza. Soup, I love soup. I'm trying to learn cook more at home, and I'm trying different type of soups. It's so cool. I never tried. Is it called a tribal? By the way, my accent,
Acai.
bowl. My accent can be a little weird. Yeah, and also if I don't pronounce your name correctly, please correct me. Okay, that's cool. I guess the food is something that connects all of us. I love food so much. And yeah, now I guess I can start showing you our laboratory. And I will first start with our like a very short our lab tour. Then I will briefly talk about our research, and then I will show you some tools that we are using in our lab, which is actually one of the most commonly used tool in our lab is microscope, and we will look some of samples under microscope together today. Okay, I will try to connect with my phone again, or maybe actually I can show my bench with my laptop. Okay, so this is my bench space. No, actually the phone will be better. Sorry for this. Let me quickly join the meeting with my phone. Recording in progress. Okay. And cool. So now, great. Yeah, this is much better. So this is my bench, and although like each labs have different research topics, the labs are actually look quite similar. Each person has their own bench. Here, this is where we do our experiments. And during we do experiments, we wear a protective equipment such as gloves. Actually, while I'm wearing my gloves, I want to quickly ask you, why do you think scientists need this protective equipments?
Not to get hurt. So what do you say, guys? To protect themselves? To protect the experiment? No. Say both? No. No, there's only both. You have to be. For both, if your hands are dirty, so you could go in the. Right, like they get germs on your hands and then you want to grow some bacteria and then your bacteria from your hands. Might get in there maybe. Well, we'll hear what you said.
Um, okay, so okay, yes, most people say it is both, and it is true. So we wear. Oh, I'm switching my laptop again. Okay. Yes. So we wear protective equipment to protect both ourselves and also for our experiment, for our sample. You are right. And the reason why we want to protect ourselves is, as you can imagine, when we are doing experiments, we are using some dangerous chemical that can be harmful for us. So we need to protect ourselves from those chemicals. And we need to also protect our samples because in our experiment, because let's say I want to mix two different solutions. I don't want anything outside to go inside that solution. So we need to keep everything clean. And one thing we use to kind of sterilize everything is ethanol. It's not surprising to all to hear that PP is also for, oh, okay. Um, okay, maybe I should. Yeah, so we use ethanol to basically sterilize everything. This is basically like a hand sanitizer, but the alcohol part. So I will clean my hands with this. And one of the most basic equipment that we use in our lab is the pipettes. So the pipettes we use to transfer different liquids and to transfer liquids. And by the way, you can ask me any questions during through chat, or if you raise your hand, we can unmute you. Okay, so with the pipette, we can set the volume we want to transfer. Here, I can hear, for example, this is 1,000 microliter, and I can here change the volume that I want to transfer. And this is something we use every day. And we also have like this kind of small pound of tubes. We add our samples in it. So this is basically how our labs, our bench space looks like. And we have other different equipments based on the experiments we want to do, such as this is a gel tank. This is we use to run agarose gel, and what agarose gel is basically a gel we use to separate like DNA. So yeah, this is how my bench spaces look like. And today I will not be wearing lab coat because we will not be really working with any dangerous chemicals, but I will still keep my gloves because I will be touching my bench. And yeah, and my laboratory, it is we are here working on something called stem cells. And before kind of introducing stem cells, I want to first a little bit talk about what cell is. And one of my favorite things to do is actually watching movies. So I kind of like using films to describe what cells are. So bear with me. So, in films, there are different actors with different roles, right? And basically, movies are created as a result of the interaction of these different actors, and each actors play their different roles. And it is very similar, actually. Our cells, our organs are formed from a small functional units called cells. And cells, different cell types, they have different functions, and by interacting with each other, they form our body and they form our organs. And stem cells, they are like an actor who can play any role. So the stem cells, they already like it's a kind of actor who already has the script, who's already familiar with the role, and they can just go in the movie and play the parts when they get the signal. So stem cells are type of cells. They can transform into, we say, differentiate into different cell types. And my laboratory, we are working on stem cells found in the skin. So today I want to kind of introduce you the skin and a little bit talk more about the stem cells in the skin. So I'm gonna share my screen to show some slides with the cartoons of skin. Oh, I think you need to enable me too. Thank you. Oops. Okay. So this is how our skin looks like. This is obviously a cartoon. Hopefully today we will also see with our own eyes how it looks like, but it has different compartments. So can you also see the mouse? Okay,
Yes.
cool. So this upper compartment is called epidermis. So this is basically what we see when we look our arms. This is the outer part of our skin, and it is called epidermis. And under epidermis, there is this part which is called dermis. And dermis has its own cell types, and also there are blood vessels and also neurons. And the other compartment that we can actually see with our eyes is the hair follicles. And similar to epidermis and dermis, hair follicles also made from different type of cells. So this is the kind of the big picture of how our skin looks like. Now we're gonna zoom into the epidermis part. So here, as you can see, our epidermis actually, when we look from outside, it doesn't seem very complex, but actually it's a very complex tissue. It forms from different layers. Here in the basal layer, we actually have our stem cells. And in the other layers, we have different type of cells. And as mentioned earlier, these stem cells in the basal layer, they can differentiate into the other cell types. So I want to ask you again another fun poll question. There is no right or wrong answer, just to interact with each other. Do you know why the stem cells might be important? Or why do you think we are working on stem cells? We are trying to understand their biology. I'm just gonna wait a little bit more. So most of you said both, and that's correct. Um, so yeah, stem cells, since they have this property of making a different cell types, they can actually regenerate a tissue. And also they play role in certain skin disease as well. So here now, I think I will start with wound healing because I think this is the one of the great examples of how stem cells regenerate a tissue. So as I think you probably all of you familiar with the wounds, and as you all know that when we have wounds, it's actually heal pretty quickly, and that's kind of awesome. And what happens is actually when we have wounds, or we have an open area in our skin, right? And we haven't talked about it, but the main role of our skin is it is actually our protective organ. It protects us from infections or like invasion of the microbes outside to our body, right? And when we have this open wounds, actually we are exposed to those infections. So when there is this wounding, actually our stem cells, they sense it and they become activated. So first they actually start to make more stem cells, and then they migrate to move to this wounded area, and then they start making different cell types. So in this case, I said, for example, stem cells are like an actors who can play any role, and when they are getting signal, they play part in the movie. Like in this case, for example, this signals is this want. When there is one, now they're like, okay, I am needed. And now they start to make different cell types. And there are actually chronic wounds. Some people have a chronic wounds. These are type of wounds it doesn't heal. So they actually need to get a treatment. Um, and there's, for example, other cases. For example, if we have a small burn, that's an easy to heal, but we can also have there big burns, like first degree burns, and those are much difficult to recover. So now, if we understand how the stem cells work, if we understand their biology, we can actually find treatments for chronic wounds or this kind of large burns, skin burns. And this is actually something called stem cell therapy. And the other thing, as you already know, is stem cells play a role in diseases as well. There are many different type of skin diseases, and one of them is, for example, skin cancers. And my lab is actually working on both wound healing and also skin cancers. So basically, what cancer is when the stem cells they go out of control. So actually, some people refer, for example, skin cancer as non-healing wounds. And the reason is that in wounds, stem cells start to make more stem cells and other cell types because they are needed, right? But in the case of cancer, even if they are not needed in that area, they start to proliferate, which means they make more stem cells, and then they form these tumors. And in some cases, the stem cells actually gain, not just stem cells, this transformed cancer cells, they gain the ability to escape from skin. And as I showed in the initial slide, under the skin there is dermis and there are blood vessels. And if they escape from the epidermis, they can actually go into blood vessel, and this cancer cells can spread other organs and form tumors in other organs as well. And how this, sorry, and how this cells are cancers, stem cells go out of control and they transform into um, tumor cells. It is still under like investigation. As scientists, we're still trying to understand how this is happening. So one thing is, for example, UV lights can cause this transformation. if we expose our skin to UV lights for a very long time, which is, the sun is emitting UV lights as well, even if we can't see them, then it may cause the transformation of the cells to cancer cells, the normal cells to cancer cells. And that's kind of why we need to wear sunscreen creams to protect our body. So yeah, do you have any questions so far? You can stop me whenever. Okay, so then how are we gonna, so it's just clearly it is important to understand the stem cells, their biology, how they differentiate into different stem cells so that we can find treatments for wounds, for burns, or for skin diseases, right? And then we need tools to study stem cells. And I think one thing that will be cool is if we were able to see them. When I look at my skin, I don't see any cell type, right? But there is a tool called microscope, which is basically magnifies the objects. And with microscopes, we are actually able to see much smaller objects that we can't see with our eyes. And here I actually want to again ask you a small question. Do you know what we can see with the light microscopes? What is the smallest thing we can see with a light microscope? Oh, right. Close. Okay.
Like, what can you see? Which organelles can you see? Good, mitochondria, what else? Excellent, good, what other organelles? Cell membranes you can see. Excellent, good job, Rosie, good for you.
You already know so much about it. It's so cool. So yeah, most of you say cell organelles. This is correct. We can see organelles with the microscope. And actually there are different type of microscope for different purposes. And we are even able to see proteins with the microscope. With powerful microscope, we can actually see proteins as well. But today with our light microscope, we will look for the skin cells. So I was watching like YouTube videos of previous like EDU events, and I find this very cool link that I want to share with you to kind of understand the scale of the microscope and how much we magnify the objects. Oh, okay. So, okay, we start with the coffee bean. We can see the coffee bean with our eyes as well, and we can certainly magnify it even more with the microscope. Then with higher magnification, we can even see now grain of salt. And our microscope is very powerful. They can even magnify unicellular organisms such as amoeba. And today we will actually look for skin cells. So as you can see, skin cells is actually only 30 micrometer in size, and coffee beans, it is in millimeter range. So we will be really, really, really magnifying very tiny cells and looking under microscope today. With that, I think we can go to our microscope room. I'm just gonna mute myself while I'm going there, and I need to carry my laptop. And meanwhile, if you have any question, please free to write the chats. Okay, I'm back, sorry. So, okay, I will show you the microscope room. So our room is dark because here we have light microscopes, and that's why basically they are working with emitting lights on our samples. Therefore, we don't want any external lights. That's why it is dark. And with my phone, I will try to show you the room. Okay. So, so this is our microscope room. We have different type of microscopes. So this is the simplest one. This is we only use to dissect this skin, like separate the skin tissue. This doesn't have very high magnification power. And today I'm gonna show you this microscope. And this is a light microscope. We call it fluorescent microscope. And first we can start with, maybe I can start with kind of showing you the parts of this microscope. So this part is called stage. This is where we place our sample. And this is eyepiece. This is where we look our samples. And here, as you can see, we have these lenses. So this is called objective. And these are basically what magnifies our sample or our specimen. So, okay, if we can see, now, can I move this? Okay, so here, as you can see, there are actually different type of objectives. So each these objectives has a different magnification power. So for example, now, this is turned into the 10x one. So I will start showing you with the 10x one, and then we will move to higher magnification to see the power of the increasing the magnification of the lenses. Oops. Okay, now I will grab my, oh, I dropped my, sorry. Okay, so I will start with showing you our skin samples. Maybe I will switch into my laptop again. Okay. Yes. Now I will show you, sorry, some of our skin samples on how they look, basically. So, okay. This is slides. Maybe I can turn, is it working better? Okay. So this is slides. We basically put our samples on these slides, and you can put whatever you want, like you can put any samples that you want. You can put blood droplets, you can put plants. In our case, our specimen, our sample is skin tissue. So I think it is difficult to see with camera, but here you see these lines. This is actually where our skin is right now on this slide. And so this is actually a technique that we use in lab called sectioning. These are skin sections. So what is sectioning is, let's think an apple. Apple has a 3-dimensional volume, right? It has length, it has depth, and it has breadth. And you can image an apple as it is in a more 3-dimensional space. And the other way, what you can do is you can make a very thin slices of apple. And this is basically the same. You can image the whole tissue as more in a 3-dimensional form, or you can make a very thin slices, what we call this section, and image more in a 2-dimensional form. And today we will look more 2-dimensional images, but then at the end, I will try to show you also the difference between how it is look when you image the whole tissue versus the sections. So again, this is where our samples are now, and I'm gonna put this to the stage to image them. Okay, so this is our microscope. Um, okay, so okay, I'm gonna put my slide here. And the lights, it will be emitted from these objectives on my sample. And this is basically how this lenses magnifies those samples. Okay. And basically using this thing, it's like a joystick. I don't know if can you see it? Yes. A joystick, we can move our slides. Okay, and our microscope, it is actually connected to computer, so we can see our images on screen like this. Okay, let's start. I will again switch to my phone. Okay, so you can see the room right now, right? My phone, it looks good. Great. Okay, so now let's start with looking our skin sample. By moving this stage, I will find my tissue. And here we can actually get live images. Oh, actually, need to make this bright field first. So first we'll use a bright field, which is basically a normal light illuminating our sample. Okay, so I will actually use the eyepiece first because it will make it better. So these are the eyepiece, and I can also see the samples through eyepiece.
So Iram, would you be able to explain a little bit of what's happening right now?
Oh, yes, sorry. I was so
No worries.
right now I'm trying to find my samples on the slide. So my sample is located in a certain place. So by using the ice pieces, I'm gonna find my sample. And this is basically the light, how the light pass. It shows the path of the light. Here, for example, you can see it pass. Okay, it passed through the objective, and here I can change to versus eyepiece. This is the place we look the samples, or through the camera. So I can switch this to camera, and then here I can choose. And when I see live, I will be start seeing on the screen our tissue. So this is 10x. So this is our skin. We can already see it a little bit better. But as you can see, we can't really say which part is what. Like I can say this is actually epidermis, and I know this part is dermis, but it is actually very difficult to understand. And we can go higher magnification, so let's do that. And here actually I need, so this is actually something we call help us to focus the image. So when I move it, as you can see, image become more clear. So this is basically something we use to move the stage. Okay, as you can see, when I choose to higher magnification objectives such as 20, this is 20x, now our tissue gets bigger and I can see much better. That's like, this is, here there's like epidermis and this part is dermis. And I think here there is actually growing hair follicle. But as you can see from the image, it's not very clear. We can't really detect the details, and it is like we can't actually see the cells. So actually, what we do to be able to detect the cells is using this technique called immunofluorescence staining. So today, now I'm gonna again switch to my laptop and show you some slides to explain how immunofluorescence staining works. Okay. So our cells actually have these molecules called proteins, and proteins has different size, different shape, and different function. For example, there are some proteins that provide a more structural support, for example, give cells their shape. And there are other proteins more help, for example, the chemical reactions to occur. And some proteins, they are found in every cell types of the skin or many different cell types. And there are some proteins, it is very specific to certain cell types. For example, protein A can be specific to only stem cells, but not the other different cell types of the skin. Then what we can do is we can actually use those proteins to detect those cells. So what we do is actually we treat those cells with a molecule called antibody, and these antibodies, they actually recognize proteins. So this antibody is specific for protein A. We can also have another antibody specific for protein B. And these antibodies, they are attached to fluorophore. This is basically, you don't really need to remember any of these names, but this is basically a molecule that when you illuminate with light, it's actually this fluorophore excites light itself, and it has different fluorophores, has different colors. Here, for example, it can be green, or it can be red, it can be blue, and by using different colored fluorophores, we can detect different proteins. So we can detect different compartments of tissue and different cell types. And now actually, in order to see our skin compartments, we're gonna look for some immunofluorescence stain samples. Okay. And now I am taking my stain sample. So this is again, I'm again using the slides. And now I'm gonna turn the slide off too, because now we will start emitting different color of lights on our sample. Okay,
So is this the same sample that we saw before, just with different colors associated with it?
Yeah, so it is like again a similar skin tissue, but this time we treat them with the antibody, these molecules with the fluorophore, this colorful text, so that we can see the different parts of our skin tissue. Okay, so let me switch back to my phone. Okay, now again, I will put my slides on stage. So the first thing I would like to do again by using this joystick, I will try to find my sample on my slides. And here, this time I will choose a color because now my sample is colored with the treatment of those antibodies. Okay, so let's see. Also, I actually need to normally turn off the light in the room too, but I think it will be fine. Okay, so now I found my sample. Now I need to focus my sample again using this thing. This is basically moving our objective up and down. Okay, so here this is oops. So now we can already see in more details, although I am not sure if you see very clearly, but so yeah, this is a staining that stains for the nucleus of the cell, which is basically the control center of the cells. It's an organelle. And here we can see hair follicles. These are going through the dermis. And now we're gonna look for different colors to detect different part of our tissue. Okay, so this green color actually detects a protein called keratin 14. I think you guys already heard about the keratin because like on shampoos, you know, people like there are things like keratin treatment for the hair. So basically what keratin is a protein found in the cells. It's actually support the structure of the cells. And actually our stem cells, they have different type of keratin than compared to other cell types. So this is a keratin 14, and now we are actually using those antibodies that detecting keratin 14. We can actually see our stem cells, and we actually, as I showed in the cartoon, they are found in the basal layer of our skin tissue. And also our hair follicles, they also have the same keratin protein, so we can also see our hair follicles as well. So this is a more an adult skin. As you can see, it has a very big hair follicles that goes deep into the dermis. Here, I will try to focus a little bit more. Now I'm going to also show you a little bit earlier stage of the skin, and we will again continue looking for different parts. Oh, I also realized it's already 4. So, okay, let me show you some more stainings so that we also have like time to chat a little bit more. Okay, here I'm also again trying to find my tissue. Okay, so I find my tissue. Again, I'm trying to focus. Okay, here you can see this is a more early stage of skin. So we have a tiny hair follicles. They are not grown as much. Here again, we see the stem cells in the base layer. And now we will gonna look for the other cell types. Here it is the more different cell types with different color. And now I can take an image and then actually see this layered state of the skin, the layered structure of the skin. And let me take a snap. Now our microscope is taking the image. Okay, so if I zoom in here, So as you can see now, we can detect with this technique the stem cells in the basal parts, and then the differentiated cells in the more in upper layers using these different colors. And here, this part is not stained because this is the dermis, and this colors that I am using, they are specific for the epidermal cells. So I can very nicely see where epidermis starts. It starts from here to here, goes to through here. And I can actually show you like more images on my slides, which will be, I guess, more clear to see. So if I share my screen again. For example, this is an image I take. So the right part is more three-dimensional, as I said, and the left part is basically what we did today. It's more in sections, it's more 2D. Here, for example, again, what we look is the stem cells in green. And here actually I stand for blood vessels of the dermis. So in 3D, you can see like you can see like the hair follicles grow through the dermis, their interaction with these blood vessels. And the 3D image is also very powerful because then you can have a more clear idea how the structure looks like.